speaker is Professor Jamboor K. Vishwanatha. He is Reagent Professor and Vice President, University of North Texas Health Science Center, USA. And he is going to share his work on cancer migration and invasion, molecular markers and therapeutic strategies. Dr. Vishwanath.
Thank you all for attending this session, and I particularly want to thank Dr. Gopinath who invited me to this meeting. I have known him for many years. Thank you very much, sir, for inviting me to this conference. So what I'm going to present today is really just focused work on one gene that we are working on in my lab. You heard from Dr. Kaul just now that it's not the primary tumor that kills you, it's really the metastasis. In most cancers, it's really the metastatic process. And the biggest challenge we have currently is how do you prevent metastasis? If we can figure out how to prevent metastasis, many of the cancers can be cured. But more than that, the quality of life of the patient can be improved tremendously. So with that, I'm going to talk about this one gene that we have isolated in our lab, which seems to be very potentially a great target for metastasis inhibition. So these are the kind of work that we do in my lab. We are looking at the role of MIN1 in metastatic progression of prostate and breast cancer, role of another gene, annexin-A2 in prostate and breast cancer progression, and some nanotechnology approaches on how to deliver some of these cancer therapeutic drugs. I cannot cover all of them, so I'll just go through the first one, the MIN1 in metastatic progression. This gene, MIN1, was originally, when we started working on it, was just an open reading frame. It was in chromosome 17 in Q12. The exact location was C17 or open reading frame 37. It's present in a hotspot for cancer. It's the C17, 1712, Q12 amplicon. If you look at the organization on the left, what you see is that, I don't think this pointer works, it does work, but on the left side, it's present in an amplicon that also contains the HER2 oncogene, along with GRAB7 and other genes. But curiously, if you look at the direction of transcription, this particular open reading frame has an opposite orientation of transcription compared to HER2 new. It's a very small protein. That's only about 12 kilodaltons, 505 nucleotides long. While when we started working on it, it was called variously C17 or F37, C35, etc. So one of my students started looking at more carefully what this particular open reading frame contains. And immediately it became obvious that this is a very, very important open reading frame. It probably codes for a protein. If you look at some of the signatures that it had, in the C terminus, you see CVIL motif. It's a very clear motif. As long as you have the C in that position, this particular protein is modified by prenylation, and it attaches to the plasma membrane. So we had a prenylation motif, but we started looking at interior of the protein. We also find another classic signature of ITAM domain, which is immunoreceptor tyrosine activation motif. And then there's other motif, CXXC motif, which is also a redox motif. So in a very small protein like this, it has multiple important domains. And I must also tell you that there are only 4 known proteins that have both an ITAM domain and a prenylation motif. So obviously this must be a very, very important protein. Curiously, you look at any normal tissue in a human, there is no expression of this C17. The only normal tissue that expresses it is the Leydig cells in the testis. That's the only normal. No other normal tissues express. But if you start looking at cancers, this is just example of breast cancer. I'm sorry, this is example of prostate cancer, but same thing happens in breast. You start looking at normal breast, you don't see any expression. In BPH, you start seeing some expression as you see from the brown staining. But if you look at the bottom panel, As the cancer progresses to Gleason grade 7 and 9, there's very high expression of this protein. We have also compared matched primary tumor versus metastatic tumor from the same patient for prostate, and you can significantly higher expression in the metastasis than even the primary tumor. But the normal prostates don't have expression at all. So this is what we knew when we started working. One of my PhD students started working on this gene, and immediately he wanted to show that this whatever the protein that results from this gene is actually membrane located, because we see the CVIL motif. So looking at both the LINCAP and the DF145 cells, the top panel shows the membrane association. You can see clearly there's very little cytosolic expression. Most of it is in the membrane. But at the bottom, we also used a technique called turf microscopy, which is a confocal microscopy in which the incident laser beam is coming at an angle. And so you collect data from just the top 40 nanometer of the membrane. So when you see those specks on the bottom, though each speck represents accumulation of protein just at the membrane, just at the inner face of the plasma membrane. Then we started constructing siRNA, and here's a migration study. So the dark area that you see on the corner is really an agarose bead in which you put in the cells, and you let the cells migrate out of the agarose bead. And the mock transfection shows that after, I think this is about 48 hours, that a lot of cells came out of the agarose bead. But as you see increasing the concentration of the siRNA, you can see the migration is completely inhibited from this agarose bead. So we knew indeed that migration is dependent upon this particular gene expression, and that by blocking the migration through siRNA, you can clearly demonstrate it. This data is just on the left side shows migration, on the right side shows invasion. So in the left side, we use the siRNA and show that the migration is inhibited. By using the Boyden chamber assay, we also overexpress C17, and you see in the right panel, when you overexpress the same gene, invasion is increased. So we have shown both by inhibiting the gene as well as by overexpressing the gene that the process of migration and invasion are affected by this gene. I'm not going through all of this data. I cannot present all of that in the short time, but here's the illustration of what happens. You see C17 ORF37 shown right in the middle at the top. Because of its prenylation, it is bound to the inner face of the plasma membrane. It activates AKT, and the phospho-AKT then activates I-kappa B, influences NF-kappa B release into the nucleus, which then activates MMP9, UPA, VEGF, which then goes out of the cell and causes tissue matrix degradation and causes migration invasion. We have actually proven every step of this through experimentation. So let me just talk about this prenylation motif. I mentioned about the CVIL motif. That cysteine in that position is invariable. You have to have that for a protein to be prenylated. There are two enzymes that modify the CVIL motif. One is geranyl transferase. The other one is farnesyl transferase. If the X is a leucine, then it's geranylated. If the X is either a serine or a methionine, then it is farnesylated. And the limited number of proteins that have this kind of a prenylation domain is shown here. Rho A, Rho B, Rho C, RAC1, CDC. The signature of all of these genes, they are involved in kind of cell migration. So MIN1 or the C17 protein is definitely involved in the migratory process. So you get cellular cytosolic MIN1 or C17. I will come to the MIN1 part. It is geranylated, and then it binds to the membrane. We constructed a number of these mutants. We either had just the plain GFP vector, or the wild type, which had the CVIL motif, or the mutant C112S. And we said, what happens to its cellular localization? in the panel A, on the top, what you see is just transfecting GFP-MIN1 cells. You can see clearly that it is membrane-bound. A colleague of mine at Duke University in Singapore has produced an inhibitor for the geranyl transferase called DU40. So if you treat these cells with DU40, the second row in the panel A shows that while the protein was bound to the membrane, you add the inhibitor for the geranyl transferase, then all of this protein is distributed now in the cytosol. Whereas inhibition with a farnesyl transferase inhibitor, shown in the third row, has no effect. So this proved to us very clearly that the CVIL motif is very important, and that cysteine is also very crucial for the protein function. More importantly, we started looking at cells that are migrating. The way we do this is we induce a scratch wound in a growing culture plate, and then we start looking at cells migrating into the wound. And in the top panel, what you see is just a scratch at 0 hours. And then at 3 hours, you can see some cells shown by the red arrows start moving into the vacant space. 6 hours, there are more of them that have migrated out. At 9 hours, there's a lot of migration. But curiously, when we do confocal microscopy of these migrating cells, shown in the colored panel there, what we see is that this particular gene, this protein, is actually expressed at the leading edge. So it is really a protein that is necessary for the cells to start migrating out. We studied this process further by looking at what is called philopodia. So for cell migration, you need two processes, philopedia and lamellopedia. These are things, protrusions come out of the cell. They're the ones that anchor to the matrix and let the cell move forward. So the top panel shows that GFP mock transfection, there's very little philopedia. But if you overexpress MIN1, as shown in the middle panel, there's a lot of philopedia. But look at the bottom row, where we just introduced one amino acid change from C112 to S112. The philopedia is completely abolished. So it shows very clearly that there is a direct link between expression of this protein and formation of philopedia, which is involved in migration. We did a lung metastasis study where we injected the cells through the tail vein of mice and started looking at accumulation of these cells in the lungs, and then looking at metastatic nodules. In panel A, what you see is mock transfection of GFP. It is just the DU145 cells injected in the mice. You see some tumor nodules formed, but in the bottom, when you overexpress this C17, you see the number of nodules have tremendously gone up. So there's a significant increase of 50-fold or so increase in lung metastasis when you overexpress this gene. Then we collected a number of lungs, and you can see the fluorescent images that also show the accumulation of this protein in the lungs. But more importantly, if you look at the last panel on the H&E stain, something curious came up, which is in the middle, when you overexpress this C17, you see that there is the tumor cells have gone to the vessel, and they have came out of the vessel, so they have invaded the tissue and formed the tumors. Whereas in the bottom, when we introduce a single mutation, the cells go to the vessel, blood vessels, but they're unable to invade out, so they're stuck in the vessel itself. So it kind of gave us a lot of confidence about what this gene is doing. With all that, we decided then, how do we, first of all, let me mention that at that point, we said we are so confident that this protein was involved in migration invasion. We proposed to Hugo Gene Nomenclature Committee, and they accepted our data. And now that all of these open reading frames and other genes, they're now called MIN1. So literature now shows this as migration invasion enhancer 1. With that said, we wanted to target it. And I mentioned that there are two very important, it's not moving. Do you know why this is not moving? Can you move the slide? It's not moving. So anyway, when he's doing that, let me just explain. So we now have the ITAM domain and the prenylation domain. We said these are great targets, especially because this protein is not expressed in normal cells at all. We could come up with therapeutic approaches. So we devised short peptides, 6 mers, against both the ITAM domain and the CVIL domain, the prenylation domain. And what we show now very clearly is that both of these peptides can inhibit the process of migration and invasion. We are determining molecular mechanism by which it's doing so. But more importantly, we are moving more into clinical translation of these peptides. We have formed D-isomers of these peptides, and they are very stable in the plasma. They are they're not cytotoxic, and we have not seen any immunogenicity because of these peptides. So we are very confident that these peptides are working. We are doing 2 things right now. One is to move it more into clinical translation. We are looking at all the PK/PD studies. We also are looking at how to cyclize these peptides so that their half-life in the plasma will increase. So that is all that's going on, and I'm sorry that you missed the best part of my presentation because we didn't do this. Where we are going on that is we are developing small molecule inhibitors against MIN1. This seems like a very unique protein that is cancer-specific, involved in the process of metastasis, and if we can successfully translate this into the clinic, I think we will very soon have a good therapeutic approach for metastasis. We have filed a patent application which is very close to getting approved at this point. So thank you very much, and I'll be happy to take any questions.